Hello students, a good news for all of you. The Nobel Prize in Physiology or Medicine has been announced. Nobel Prize 2024 in Physiology or Medicine has been announced. It goes jointly to Victor Ambros and Gary Ruvkun. It goes jointly to Victor Ambros and Gary Ruvkun. For what? For the discovery of microRNA and its role in post-transcriptional gene regulation. So the Nobel Prize in Medicine 2024 goes to Victor Ambros and Gary Ruvkun for the discovery of microRNA and its role in post-transcriptional gene regulation. This will be a very important question in your prelims as well as mains. The information stored within our chromosome can be likened to an instruction manual for all cells in our body. Every cell contains the same chromosome, so every cell contains exactly the same set of genes and exactly the same set of instructions. Yet, different cell types, such as muscle and nerve cells, have very different distinct characteristics. And how do these differences arise? The answer lies in gene regulation, which allows each cell to select only the relevant instructions. Remember carefully, it is only the relevant instructions. This ensures that only the correct set of genes is active in each cell type. So the genes are same of every cells, but only a correct set of genes is active in each cell type. Listen carefully. If you don't understand, rewind the video again and see it. You will be able to understand it better. This concept is very important. Victor Ambros and Gary Ruvkun were interested in how different cell types develop. For example, muscle cell, nerve cell. They discovered the microRNA. What is microRNA? A new class of tiny means very small RNA molecules that play a crucial role in gene regulation. Their groundbreaking discovery revealed a completely new principle of gene regulation that turned out to be essential for multicellular organisms, including human. We are humans. Humans are multicellular organisms. It is now known that human genome codes for over 1,000 microRNAs. Human genome codes for 1,000 microRNAs. Their surprising discovery revealed an entirely new dimension to gene regulation. Their surprising discovery revealed an entirely new dimension of gene regulation. MicroRNAs are proving to be fundamentally important for how organisms develop and function. Microorganisms are now proving to be a fundamentally important how organisms develop and function. This year's Nobel Prize focuses on the discovery of vital regulatory mechanism used in cells to control the gene activity. Genetic information flows from DNA to messenger RNA, mRNA, via a process called transcription, which we all know, and then on to cellular machinery for protein production. These mRNAs are translated so that proteins are made according to genetic instructions stored in DNA. Since the mid 20th century, several of the most fundamental scientific discoveries have explained how these processes work. Our organs and tissues consist of many different cell types, all with identical genetic information stored in their DNA. However, these different cells express unique set of proteins. How is this possible? The answer lies in precise regulation of gene activity so that only the correct set of genes is active in each specific cell type. This enables, for example, muscle cells, intestinal cells, different types of nerve cells to perform their specialized functions. In addition, gene activity must be continually fine tuned to adapt cellular function to changing conditions in our body and environment. If gene regulation goes RE, it can lead to serious diseases such as cancer, diabetes, or autoimmunity. Therefore, understanding the regulation of gene activity has been an important goal for many decades. In the 1960s, it was shown that specialized proteins known as transcription factors can bind to specific regions in DNA and control the flow of genetic information by determining which mRNAs are produced. Since then, thousands of transcription factors have been identified, and for a long time it was believed to be that main principle of gene regulation had been solved. However, in 1993, this year's Nobel laureates published unexpected findings describing a new level of gene regulation, which turned out to be highly significant and conserved throughout evolution. This year's Nobel Prize focuses on discovery of vital regulatory mechanism used in cells to control gene activity. Genetic information flows from DNA to messenger RNA via a process called transcription and then on to cellular machinery for protein production. Their mRNAs are translated so that proteins are made according to genetic instructions which are stored in the DNA. Since the mid 20th century, several of the most fundamental scientific discoveries have explained how these processes work. Our organs and tissues consist of many different cell types. Now, research on a small worm lead to a big breakthrough. Now, the real story is here. In the late 1980s, Victor Ambros and Gary Ruvkun were postdoctoral fellows in the laboratory of Robert Horvitz, who was awarded Nobel Prize in 2002 alongside Sidney Brenner and John Sulston. In the Horvitz laboratory, they studied a relatively unassuming 1 millimeter long roundworm, C. elegans. Despite its small size, C. elegans possesses many specialized cell types such as nerve and muscle cells also found in larger, more complex animals, making it a useful model for investigating how tissues develop and mature in multicellular organisms. Ambros and Ruvkun were interested in genes that control the timing of activation of different genetic programs, ensuring that various cell types develop at the right time. They studied two mutant strains of worms, Lin4 and Lin14, that displayed defects in timing of activation of genetic programs during development. The laureates wanted to identify the mutated genes and understand their function. Ambros had previously shown that Lin4 gene appeared to be a negative regulator of Lin14 gene. However, how the Lin14 activity was blocked was unknown. Ambros and Ruvkun were intrigued by these mutants and their potential relationship and set out to resolve these mysteries. After his postdoctoral research, Victor Ambros analyzed the Lin4 mutant in his newly established laboratory at Harvard University. Methodical mapping allowed the cloning of gene and led to an unexpected finding. The Lin4 gene produced an unusually short RNA molecule that lacked a code for protein production. These surprising results suggest that this small RNA from Lin4 were responsible for inhibiting Lin14. How might this work? Concurrently, Gary Ruvkun investigated the regulation of Lin14 gene in his newly established laboratory at Massachusetts General Hospital and Harvard Medical School. Unlike how gene regulation was then known to function, Ruvkun showed that it is not the production of mRNA from Lin14 that is inhibited by Lin4. The regulation appeared to occur at a later stage in the process of gene expression through shutdown of protein production. Experiments also revealed a segment of Lin14 mRNA that was necessary for its inhibition by Lin4. These two laureates compared their findings, which resulted in a breakthrough discovery. The short Lin4 sequence matched a complementary sequence in the critical segment of Lin14 mRNA. Ambros and Ruvkun performed further experiments showing that Lin14 microRNA turns off Lin14 by Lin4 microRNA turns off Lin14 by binding to complementary sequence in its mRNA, blocking the production of Lin14 protein. A new principle of gene regulation mediated by previously unknown types of RNA, microRNA, had been discovered. The results were published in 1993 in two articles in Journal of Cell. The published results were initially met with almost deafening silence from the scientific community. Although the results were interesting, the unusual mechanism of gene regulation was considered a peculiarity of C. elegans, likely irrelevant to humans and other complex animals. That perception changed in 2000 when Ruvkun's research group published the discovery of another microRNA encoded LET7 gene. Unlike Lin4 and LET7 gene was highly conserved and present throughout the animal kingdom. The article sparked great interest, and over the following years, hundreds of different microRNAs were identified. Today, we know that there are more than 1,000 genes for different microRNAs in humans, and that gene regulation by microRNA is universal among multicellular organisms. In addition to mapping of new microRNA, experiments by several research groups illustrated the mechanism of how microRNAs are produced and delivered to a complementary target sequence in regulated mRNA. The binding of microRNA led to inhibition of protein synthesis or to mRNA degradation. Intriguingly, a single microRNA can regulate the expression of many different genes, and conversely, a single gene can be regulated by multiple microRNAs, thereby coordinating and fine-tuning entire network of genes. Cellular machinery for producing functional microRNA is also employed to produce other small RNA molecules in both plants and animals. For example, a means of protecting plants against virus infection. So, these tiny RNAs with profound physiological importance, gene regulation by microRNA first revealed by Ambros and Ruvkun, has been at the work for hundreds of millions of years. This mechanism has enabled the evolution of increasingly complex organisms. We know from genetic research that cells and tissues do not develop normally without microRNAs. Abnormal regulation by microRNA can contribute to cancer. The mutation in gene coding for microRNA have been found in humans, causing conditions such as congenital hearing loss, eye and skeletal disorder. Mutation in one of the proteins required for microRNA production result in DICER1 syndrome, a rare but severe syndrome linked to cancer in various organs and tissues. So this was all about the Nobel Prize story. This story is very deeply inspiring so that more research can be done, and the complete coverage of story is in this video. If you are unable to understand, please comment in the comment box. I will make you understand the things. You can join my 3-day IAS Breakthrough Blueprint Masterclass by clicking the link in the description box. I will come with more videos. Please ask for more. I will come with more videos. My mission is that you all should succeed. I wish you all the best.